Good afternoon, everyone. Welcome to BioVanguard Cell Talk presentation. For today's presentation, Visalakshi KB is the presenter, and the topic she selected is neoantigen vaccine strategies in cancer treatment. Visalakshi KB is working as research assistant at Renix Life Sciences. She is working on multiple projects. Some of her projects include identification of biochemical properties of amino acids, gene maps, success of clinical trials, and she is also involved in writing content for social media posts. She holds a master degree in bioinformatics from Alagappa University, Tamil Nadu, and completed her bachelor's degree in biochemistry from Sri Adi Chunchanagari Women's College at Kambam, Tamil Nadu. She has also completed her internship at Indian Institute of Science, Bengaluru, and thus gained valuable experience. Her goal is to constantly gain in-depth knowledge and apply in research projects and also write content as part of scientific writing in the field of bioinformatics. So I welcome Visalakshi to give your presentation.
Thank you, Ma, for your nice introduction. Today, I have selected the topic as neoantigen vaccine strategies in cancer treatment. Coming to the introduction part, neoantigens are tumor-specific and arise from somatic mutation in cancer cell. The key features are highly tumor-specific and highly immunogenic. Neoantigens are arises from genomic mutation and translated into the novel protein. These are recognized as novel target for cancer immunotherapies. Types of immunotherapies. There are two types of immunotherapies. One is active immunotherapy, another one is passive immunotherapy. When it comes to the active immunotherapy, it stimulates the patient immune system naturally to attack the cancer cell by immune cells like T and B cell. When it comes to the passive immunotherapy, it provides preformed immune compounds without activating the patient immune system. It relies on immune compounds like monoclonal antibodies, cytokinins to target the cancer cell. How to recognize the tumor neoantigen by CD8 plus T cell. These tumor neoantigens are novel peptides present on the surface of the tumor cell. Neoantigens are recognized by CD8 plus cytotoxic T cell. These cells are key for the anti-tumor immunity. It is recognized neoantigen present on the tumor cell via major histocompatibility complex class 1 molecule. How to process and recognize the tumor neoantigen. Step 1, first we have to generation of the neoantigen. Somatic mutation in tumor cell that produce the mutant protein. These proteins are processed into the peptide by proteasome loaded onto the major histocompatibility complex class 1 molecule in endoplasmic reticulum. In step 2, presentation of neoantigen. These tumor neoantigen peptides are present on the surface of the tumor cell, which bound to the major histocompatibility complex of the class 1 molecule. Step 3 is CD8 plus T cell activation. In T cell receptor, CD8 plus T cell that recognize the neoantigen as major histocompatibility complex of class 1 complex that interacts between the co-stimulatory molecule of the stimulatory molecule that enhance the activation. For example, CD28 on T cell with B7 on antigen presenting cells. When it activates the CD8 plus T cell that enhance the proliferation and differentiation into the effector of T cell. How it kill the tumor cell? Effector of CD8 plus T cell that kill the tumor cell directly without affecting the healthy cells. Presentation that approach for identification of neoantigen. First, to identify the patient-specific neoantigen that include the advancement of genomic sequencing, bioinformatics, and immunology. The key approaches are genomics and transcriptomics and bioinformatics pipeline and immunopeptides. This diagram represents that the source of the potential neoantigen like SNV, indels, translocation, and alternative splicing, which transfer into the whole exome sequencing that identify the non-synonymous mutation by using cDNA library screening. Then it moves to the sequencing of the tumor versus non-tumor DNA. Then it will unfilter the neoantigen candidate that predicts the in silico epitope prediction. In immunopeptidomics, that select the selection of neoantigen candidates, which predicts the immunogenicity of neoantigen. Uh, it produces the synthetic neoantigen in lab. That neoantigen synthetic will be given to the personalized cancer patient to activate the immune cell. When it comes to the bioinformatics pipeline, the whole exome and whole genome sequencing will be done by the sequencing machine, and the generated FASTQ file will be given for the quality control for adapting adapter and lower quality base trimming by using the tool FastTP, Trimomatic, Cut, Adapt, and PrintSeq. Then the alignment which represent to the reference genome BAM file by BWA and Novo align. Then post alignment processing will be held by using the sort and remove the duplicate reads by SAM tools and Picard tools. Then indel realignment by Getch pathway. Then base quality score recalibration will be done. Then after this, moving forward into the variant calling to identify the somatic mutation and germline mutation like SNPs and indel by using VCF file. The tools are VARSCAN2, mutate2, and haplotype caller. When it comes to the RNA-seq data analysis, RNA-seq of the FASTQ file will be given to the adapter, and low quality read trimming will be cut by using the FastP, Trimomatic, Cut, Adapt, and Print Skin. Then the alignment with the respect to the reference genome BAM file by using the star Bouty Topat is the tool which will be align the reference based genome. Then get the read counts by FPKM, fragment per kilo base for each gene seq by using the HT-seq. Then the process of variant calling information is added. Add the RNA expression of FPKM data and will be annotated by the variants like ensemble, VEP, Unaware, and SNP tool. Then after that, they have done a HLA typing by using the HLA minor or HLA reporter. Then HLA list will be given to the neopeptide prediction by using the PVAC tools or MUPAC tools by using the predicted HLA peptide binding affinity. When it comes to the types of cancer vaccine, there are many types of cancer vaccine, but particularly personalized vaccine will be focusing on genetic based approaches, mRNA vaccine based approaches, protein peptide based approaches, viral bacteria based approaches, cell lysate based approaches. These are my reference. Here I have taken about the overview of the neoantigen of cancer vaccine. Then the part 2 will be continue from this. Thank you. If you have any question, you can ask me.
Thank you, Visalakshi, for the presentation. Now the panel is open for any queries. Okay, I don't see any questions. Okay. So thank you, everyone, and I hope to see you all again in the next presentation.